So we were interested in acute lymphoblastic leukemia patients who are treated with CD-19 directed immunotherapy, and we focused on patients who relapsed after this treatment and they lost the target antigen. That means that they were CD-19 negative at relapse. And we were interested in the mechanism, how this CD-19 negative phenotype is caused. So we performed exome sequencing to identify mutations which could be responsible for that immunophenotype. And we checked the CD-19 gene in 17 patients which we had in our cohort. We expected to find quite some mutations, but surprisingly we only found mutations in 5 out of the 17 patients. I say surprisingly because in quite a similar cohort of patients who were treated with CAR T cells, which is also a CD-19 directed therapy, they identified CD-19 mutations in all of them. So the fact that we only found mutations in less than 1/3 of a patient surprised us. So we looked into other genes because exome sequencing makes it possible. And what we discovered was that some groups of genes which are responsible for the calcium transport and for cholesterol synthesis were mutated in quite some patients. I think it was 5 or 6 patients from the cohort. So we made a hypothesis that maybe this could be responsible for the CD-19 negative loss. But of course we don't know it. We have to investigate it further. Probably on bigger patients cohort or do some knockout experiments to confirm that these mutations which we discovered can cause that phenotype. And it would be really great to explain that because we need to understand the mechanism of the CD-19 loss, because we of course need to prevent that. Maybe that would help to prevent the relapses as well. Yeah, next is probably those knockout experiments, or we unfortunately don't have RNA from the patients, so we cannot confirm that it's not expressed. So we will probably do the knockout experiments, and we're planning to include more patients to see if it's true also in other ones.